I'm Daniel Levine, and this is the Bio Report.
The emerging understanding of the role the microbiome plays in wellness and disease is opening up a large number of potential therapeutic targets in the millions of genes that drive the microorganisms that live within the body. Eligo Bioscience is developing a new class of precision medicines that use gene editing to address the expression of pathogenic genes in the microbiome to treat not only infectious disease, but other conditions such as inflammatory diseases and cancer as well. We spoke to Xavier Duportet, CEO of Eligo Bioscience, about the company's use of synthetic DNA to target bacterial genes, its platform technology, and the wide range of conditions that can be addressed through this approach. Xavier, thanks for joining us.
Thanks for having me.
We're going to talk about the microbiome, Eligo Bioscience, and its use of synthetic DNA to develop in vivo therapies that target bacterial genes in the microbiome underlying human diseases. How well understood is the role of the microbiome in human disease today, and how broad a set of conditions might be addressed through modulating bacteria there?
So the role of the microbiome in human diseases is indisputable. Microbes live all over us, on our skin, in our guts, in our lungs, and they play definitely a major role in disease, but in health too. And so the microbiome revolution started about just over a decade ago, and there is still today so much to discover, to understand about how these trillions of resident bacteria interact with the host. And so during its first decades, the field really focused on establishing big data sets that enable to highlight correlations between specific microbial consortia and disease settings. And so today you can find publications for almost every disease that describe an increase in this or that bacterial species, or a decrease in this or this other bacterial species in patients compared to healthy cohorts. However, these are only correlations and may actually only be the consequence and not the cause of the disease. But today, the field has now significantly matured, especially in the past few years, with the emergence of much deeper sequencing methods, for instance. And this enabled us to really focus on identifying how specific changes in the microbiome at the strain level contribute to the disease development. And so once you have a precise understanding of what bacterial strain, what bacterial gene influence the host, then one need to develop precision medicines that can modulate the microbiome in a targeted way, with really in a precise way, to have the potential to be disease modifying. And then the therapeutic opportunities are really vast, from dermatology to oncology, autoimmunity, cardiology, CNS, inflammation. Modulating the microbiome with precision has definitely a bright future.
You're developing a new class of therapies you call Eligobiotics. What are Eligobiotics?
So indeed, Eligobiotics are definitely a first-in-class modality, or a first-in-class gene editing modality. And they enable us actually to bring this required precision, we're talking about, to the microbiome modulation. So our Eligobiotics achieve that through the precise targeting and the specific gene editing of the resident bacterial genes. So the genes of the commensal microbiome that are engrafted, that evolve symbiotically with the host, and these genes that have validated causal link to significant disease. And so, thanks to it's really thanks to the big and powerful synthetic biology platform and capabilities that we have that enable us to create therapeutic delivery particles that are both specific to the target bacteria as well as to the target gene within the bacterial species or the strain. And so once we've delivered with this particle a DNA payload, these DNA payload, can express a gene editing enzyme of choice, and therefore can either remove, inactivate, or add genes to the targeted bacteria in order to alter its genetic makeup, and as a consequence, positively modulate the host response. And so, very importantly, we don't have any concern in terms of target, because human cells are not transduced as opposed to all the other gene editing approach. The particles only target bacterial cells. And so these oligobiotics and their unique abilities really enable us to deploy the whole suite of gene editing capabilities to a unique set of targets and address unique therapeutic opportunities that are currently not addressable by any other technologies. And at first, with these oligobiotics, we have decided to focus on high-value indications in 3 fields: in autoimmunity, dermatology, and oncology. And we have specifically chosen targets for which the causal relationship is well established, so beyond correlations, and for which the clinical path are actually de-risked by the ability to stratify patients who are the most likely to respond to the treatment.
I know you're looking well beyond infectious disease, but from a point of view of antibiotics, it would seem there's a big opportunity to go after targets that have traditionally been treated with those. Mm-hmm. What are some of the advantages oligobiotics might have over antibiotics?
So when we started Eligo, we had indeed just published 2 Nature Biotech papers where we made the first demonstration that the delivery of a CRISPR system on a DNA payload to a bacteria could be used to destroy bacterial genes and at the same time kill the bacteria carrying the genes, both in vitro but also in vivo, and numerous animal models with their own microbiome. And so at that time we had invented what we called sequence-specific antimicrobial. So basically the most precise antimicrobial to date. And with these, we can indeed kill specifically strains that harbor pathogenic genes, deleterious genes, and leave the rest of the microbiome completely intact. So, this technology, indeed, that is like a super precise antibiotic, really led to the grant of the landmark patent in the field of microbiome gene editing, the really earliest patent in the field. But when we look at and when we think about antibiotics, really precise antibiotics, so it's clear that, as stated earlier, the microbiome is as much associated with disease than it's with our health. And so it's clear that traditional antibiotics, beyond the threat of fueling the rise of antibiotic resistance globally, there are modalities that are really effective, but they're not specific at all. And therefore, they will kill the pathogenic bacteria causing the infection, but they won't spare all the beneficial bacteria from the microbiome. And so definitely more precise alternatives are definitely needed. Drugs that can only target the bacterial species causing the infection, sparing the rest of the microbiome. And for this, there are actually a couple of new modalities that work very efficiently. I mean, used in terms of new modality, very actually old modalities, but that are becoming a bit trendy now with a couple of companies pushing them forward. It's phages, for instance. So the phages that are the replicative viruses that prey on bacteria. And so once they infect the bacteria, these phages, they replicate inside the bacteria. They employ very sophisticated strategy to create actually as many progeny particles as possible by degrading bacterial genomes with nuclease. And some companies are even today engineering these phages with CRISPR, with the goal to increase their potency of this bacterial killer that are already extremely potent. But when you look at an oligobiotic that is equipped with a CRISPR payload, it's not a phage, it's not an antibiotic, and they're much more precise. So they're more kind of highly precise microbiome modulator that can kill a specific strain within a bacterial species, so really a subpopulation of a bacterial species, a bacterial member of your microbiome. And so therefore, we do not really focus on infectious disease because, why use something really complex when nature has already made the perfect predator? We really go one step further. And so we focus on disease where you do not want to kill the whole species, but instead you really want to kill just the specific, bad member of the family that is causing trouble. And so this really opens up therapeutic opportunities beyond infectious disease, of course, beyond preventing infection or treating infection, for which the current the phages or engineered phages are already really well suited for. But we're looking at opportunities where precision really brings a unique edge that no other technology can address. And I'd like to add that the CRISPR killing is really just one of the application of the platform. We can also edit the bacterial genes without killing the bacteria, or even add therapeutic genes to transform the microbiome into a local drug factory.
What does it take to manufacture an oligobiotic, and can this be done cost effectively relative to the cost of an antibiotic?
Yeah, so that's a great question. So, we're not comparing ourselves, to antibiotics. Yes, to answer your questions at the cost level, we have put in place and we've developed, really interesting and sophisticated strategies to really optimize the fermentation process, because we produce our particles via classical fermentation, bacterial fermentation, and also downstream processes that really enable us to produce particles at very high titers with really high purity and at low cost. They're not small molecule, they're biologics, but for biologic, we can produce them at really low cost. We've actually recently de-risked the CMC of the whole platform by releasing our first 100-liter GMP-like batch. And so, to go back to the comparison with antibiotics, I think we've been really excited in the past a year, because as we're advancing different programs, we got really positive feedbacks from a number of market access experts that are, valuing our modality, expecting evaluation of and pricing of the modality at a price similar to a high-price biologics, and not all small molecules like antibiotics.
There's been a lot of concern about the growth of antibiotic resistance. There's been a lot of concern about the growth of antibiotic-resistant bacterial strains and the lack of new antibiotics to expand the arsenal. To what extent could oligobiotics address that gap?
So I think, we are, one of the ability of Eligo would be to not go after, again, infectious disease, a treatment of infectious disease. One avenue is potentially the precise removal of antibiotic-resistant genes by using our CRISPR approach to prevent the spread of these genes. Though right now, this is not an area where we're mostly focusing. We're really targeting a disease where they're not associated to infections.
I want to talk a little about the platform technology here.
Yeah.
It seems like the microbiome has a lot of complexity you need to unravel. What's the process for developing oligobiotics? How do you determine not only the bacterium of interest, but the specific genes you need to target?
This is I think the $1 million question. So we've built our pipeline with indications for which there is a strong mechanistic understanding of the disease with a defined biology. And luckily for some disease targets, the genes that we want to edit, destroy, or add, they're already known and described in the literature, which makes the work a lot easier. But now we have in parallel, and also to get an edge beyond the technological edge, but also an edge on the targets. We have already initiated a number of undisclosed collaborations with KOLs in our therapeutic areas of interest, with whom we've actually already filed co-IP on novel targets. And we want double down on a specific therapeutic area for which we have actually started our own internal target discovery effort to build an enduring pipelines of novel and proprietary targets.
What is the GEM platform, and how exactly does it work?
Right. So the GEM, our GEM platform, GEM stands for gene editing of the microbiome. And so the platform is a high throughput platform that is backed both by really strong IP, but also, and this is really our expertise at Eligo, we combine synthetic biology, protein engineering, automation, and bioinformatics to really enable us to, as soon as we have identified a gene that we want to target and the bacteria that carry this gene, we can design, build, test highly optimized delivery particles and the DNA payloads for these bacteria. And so we have actually now achieved that multiple times for intractable bacteria, bacteria that are keystone members of the microbiome, and bacteria for which actually people have been trying to deliver plasmids, have been trying to modify them for 20 years, but have not succeeded. And really the platform we've developed enables us to do that, enables us to develop de novo genetic engineering toolbox for species that are not the typical species. A lot of companies work with E. coli, Staph aureus, the pathogenic species that people have been working on for a while and are associated with infections. But as soon as you go into the more real commensal microbiome, then it's a completely new world. And for these bacteria, you need to have methods and techniques to know how to deliver DNA and to know how to create these particles that can deliver the DNA. And typically, I think the best example we can use to illustrate the power of the platform to create these delivery particle is the partnership with GSK on the acne program, where the bacteria that we're targeting in this acne program, it's a bacteria that people have been, we know it is central in the disease, in actually not only acne but a number of skin disease and skin health too. But nobody has. It's one of the hardest bacteria to work with. It grows extremely slowly. Nobody managed to get DNA into this bacteria, exogenous DNA. And in less than 18 months, we actually did it. We developed particles to deliver DNA via this GEM platform into these bacteria. And this is what got GSK extremely excited. And so now we're deploying all these techniques with the strong IP on a lot of other keystone bacteria of the microbiome to really target really high value and disease programs.
I do want to talk more about that alliance, but before we do, one of the things that's interesting to me is that you can use oligobiotics to not only kill pathogenic bacteria, but also modify bacterial genes to remove a pathogenic gene sequence but leave the bacteria intact, as well as delivering therapeutic genes to bacteria to modulate a disease mechanism. Might you want to modify a gene but leave the bacteria otherwise intact?
That's a great question, and we're super excited. We started indeed with CRISPR to kill a bacteria by destroying its genome. But more recently, we actually got a really broad patent on that new gene editing approaches. We have demonstrated in vivo that in animal model that we can, with a single administration of oligobiotics orally, we can actually modify the genome of a resident bacteria in the gut, for instance. In less than 8 hours, modify 100% of the whole population, one or multiple nucleotide. And this is super exciting. We do not kill the bacteria. We can really do gene editing, base editing inside the microbiome in situ. Why is this so exciting? In a number of diseases, the bacterial gene that drives the disease is actually carried by the whole bacterial species, the whole family. But these bacteria, because they are this family, this bacterial species, because it's a symbiotic bacteria, you cannot you cannot remove it. You cannot kill it. If you try to kill it, it will come back. So the only way to inactivate the deleterious genes, which is driving the disease, is actually not to remove the bacteria because it's impossible, but to genetically modify the gene. And for instance, change one base pair that will modify the conformation of the protein that interact with the host, trigger inflammation, modify one amino acid in a peptide sequence that is actually immunogenic. And so, when you think about the possibility to modify a gene in situ in the whole bacterial family, then you have a lot of application that start to be addressable. And so, the ability to do this without killing the bacteria is really something that now we're betting. We're advancing a lot of programs on because we're actually seeing that. Yeah, for a variety of disease, it's the whole family that carry the deleterious gene, not a subpopulation that you could eliminate, but it's really the whole species that you cannot eliminate, so you have to modify its state.
At the same time, is there the potential to turn bacteria into drug factories, as it were, and program them to produce a therapeutic protein?
Yes, of course. So, really, our delivery particle, they can deliver DNA of any size. We're not really, we're not restricted by the payload size of what we can deliver in the bacteria. So we can deliver, gene editing enzyme, CRISPR, base editor, prime editors, but we can also deliver information for the bacteria to express any type of molecules you want. And I think, this is actually really differentiating us from the whole probiotics or engineered probiotics that are used to express therapeutic proteins. The main differentiation is that our technology really enables us to leverage the existing microbiome, the bacteria that are already well engrafted in the gut, on the skin, in the lungs, and so really, indeed, turn them into local drug factories. By delivering this DNA payload that contain the information for them to produce or secrete, display a molecule locally. And this is really important because one of the major issues today that exists with engineered probiotics, for instance, is that these exogenous bacteria are feeding to the patient or applying on their skin. They have to fight with the microbiome residents, the bacteria that are there engrafted that have lived in symbiosis with the host forever. And so these exogenous engineered bacteria, they are actually very often kicked out of the microbiome shaded. And so, when we started with CRISPR, then base editor, and we thought, well, given what we can deliver, and the current constraints of the engineering probiotic space, let's add a third approach to our gene editing toolkit. And so now we have actually proven already that we can, deliver genes in gut bacterial population and produce at significant level already immunomodulatory molecules, such as nanobodies or cytokines, for instance, in the guts via this approach.
Last year, you mentioned that Eligo entered into a research and option agreement with GSK to develop a CRISPR-based approach to treating acne. This is a deal worth up to $224 million in license fees and potential milestones, as well as royalties on sales. Why did Eligo do this transaction?
I have to say, since the beginning, so we're in discussion with a number of pharma, and since the beginning with the GSK team and the vaccine team, we've been really, really well aligned, and I think they've really understood really rapidly the potential of the approach. Acne is a really big market, and I think for us, partnering with pharma at this stage, the pharma that and with whom we were completely aligned on how to move forward the drug. It was an internal program. They were highly interested in acne. I think it was just a very good match, and it is still a very good match.
How well treated is acne today, and what's the role of bacteria in the condition?
So, acne is actually most of the acne drugs available in the markets. They only provide symptomatic treatments, and they really don't cure the condition. There is also no treatments today in the market that address this acne-associated dysbiosis skin bacterial flora. So the fact that there is a subpopulation. So there's one bacteria in our skin. I go back high level to the role of the bacteria, but there is this. Our skin is populated with numerous bacteria, but there is this one bacterial species called Cutibacterium acnes that represents about 90% of all the bacteria we have on our skin, mostly on the oily surface. So, the face, the chest, and the back. And what has been demonstrated in the past years is actually, so this bacteria is critical for the skin health, really, really critical. It fights against other pathogens. It helps produce antioxidants. It helps with the hydration of the skin. But there's a subpopulation within this bacterial species that does carry a set of genes that lead to the secretion of pro-inflammatory molecules, and when expressed in a pilosebous unit in the hair follicle, these pro-inflammatory molecules lead to skin lesion, a pustule and skin lesion. And so today there's no treatments that can address this specific dysbiosis that can specifically remove target the pro-inflammatory genes in this subpopulation and remove this subpopulation. And so, because treatments are not precise today, they're not targeted to the real cause. They actually have poor adherence because of all the side effects and their ineffectiveness. And I think today the most effective drug against acne is a drug called oral isotretinoin, Accutane or Accutane. It's very efficacious, but has to be taken with care. It has a black box warning, iPlate registry, and quite a lot of administrative burden for the physicians, and significant compliance burden for patients over 6 months. This is really a drug of last resort, and there is a huge potential, I think, and unmet need before we needed to getting to this drug of last resort. And I think in the acne field, there's not been really a lot of new drugs in the past 10 years, 15 years, maybe 20 years. And so, now that the role of the bacteria has been well identified, we're quite excited that we can target this causative mechanism and try it and cure a patient one day.
You have a candidate EB005 in development. What is that, and how does it work?
So basically, it's a delivery particle that delivers. So it's a topical treatment. It will be a topical treatment that deliver a payload, a DNA payload in the whole population of Cutibacterium acnes, this skin commensal bacteria. And this payload expresses a CRISPR nuclease that is programmed to target and destroy this subset of pro-inflammatory genes that are only carried by this the subpopulation that is associated to acne in acne patients. And so by destroying these genes, the nuclease will actually, single cuts in these genes actually lead to the immediate, in terms of, in a couple minutes, to the degradation of tens of thousands of DNA base pair around the cut sites in the bacterial genome. And the bacteria cannot repair the genome, their genome, as opposed to eukaryotic cells, basically. And this will kill very specifically the bacteria that carry these genes, and therefore really rebalance the microbiome by removing this pro-inflammatory population from the skin of these patients, and therefore preventing any further lesions.
Where are you in development, and what's the development path forward?
This is a question we often have, and I guess you know the answer. Unfortunately, this is contractually confidential, so I cannot really talk about that.
Is there some decision point that GSK, though, has to make a go or no go on it?
So we are in. So here, Daniel, I actually don't know if I can answer that neither.
Let me ask you about another candidate you have in development, EB-101, which is being developed for the treatment of acute inflammatory myocarditis and prevention of chronic heart damage. What is acute inflammatory myocarditis, and how does the condition manifest itself and progress?
Yeah, so this is our lead program in autoimmunity. Super interesting program, and really excited about it. Myocarditis, so it's a severe inflammation of the heart that can be triggered by infections, viral infection or bacterial infections, that leads to cardiac dysfunction and increased mortality. In genetically predisposed patients, actually there's a phenomenon called bacterial mimicry that worsens both the acute phase of the myocarditis and also the subsequent lifelong cardiomyopathy, which occurs in approximately 50%. Well, it's 47% of patients. So very disease that has significant burden for patients, given that most of these patients are actually between 25 and 40 years old.
And what role do bacteria play in the condition?
So that's actually so that has been understood and discovered just a couple years ago, and it really demonstrates how a gut resident bacteria, which is called Bacteroides theta-iota omicron. It's a long name, so I'll call it B theta. It's easier and simpler to follow, and it's also easier for me to repeat. So how this B theta, this gut commensal, actually drive worsening of the disease and also drive long-term sequelae of the disease. And it's one of these diseases in which actually it has been demonstrated that these gut commensal, B theta, actually carry exact copies of a peptide mimic of a human autoantigen. And this peptide mimic expressed by this gut commensal actually worsen autoimmunity. How does that work? Bacteria are continuously sampled by gut immune system. So bacteria in the gut are continuously sampled by gut immune cells. And so in these genetically predisposed patients, some bacterial peptide mimic, and here this bacterial peptide mimic from B theta, can actually induce activated Th7 cells that are then able to recognize both the bacterial peptide mimic, but also the host autoantigen. And so during myocarditis, what happened is that because of the severe inflammation of the heart, the damaged heart cells actually release protein and peptides, and one of these peptides is a myosin heavy chain 6 peptide, which is an autoantigen that is actually the exact same peptide than the B theta peptide, and therefore it attracts the gut-derived Th7 cells that have been previously induced by the bacterial peptide mimic, and therefore triggers autoreactivity, and these cells, these Th7 cells, therefore increase inflammation and worsen the whole heart inflammation in the heart disease.
What is EB101 and how does it work?
So it's really EB001 is again a precision medicine. It's a vector. It's a delivery vehicle that's here. It's not topical treatment, but we're targeting a gut bacteria. It's going to be administered orally, and it will deliver in B theta. This gut commensal, not a CRISPR this time, actually, because all the B theta in the gut do express this peptide mimic of host autoantigen, the MY86 peptide. And so it will deliver a base editor that will actually do a very precise editing, so in situ editing of the gene sequence, the genetic sequence that code for the peptide mimic, and modifying one amino acid of this peptide mimic, leading to a bacteria that will have secrete a peptide which is not a mimic anymore, and which therefore will be non-immunogenic peptide. This modification of the bacterial genome is stable. It doesn't create any fitness defect to the bacteria, and therefore, because the bacteria will not express any more immunogenic peptide, it will no longer be able to worsen the disease, and therefore it will lead to an alleviation of one of the major driver of this autoreactivity, and therefore decrease help the symptoms to be alleviated much faster and prevent long-term sequelae. And I think something really important to say is that this approach is really an approach that's is disease modifying because it addressed the cause of the disease. And if you look at current treatment today, they're only symptomatic, and so we're really excited about this approach that definitely ought to be tested in humans very soon, I hope.
What's the development status of that and the path forward?
So we're now advancing, in preclinical work, and so advancing towards an IND. And hopefully, by the end of by the end of 2024, being able to being ready to go to humans.
Given the broad potential for the technology you've got, how are you prioritizing the indications you're pursuing?
Now we're prioritizing them, according to. We need strong target validation, strong market opportunity, and high unmet need and speed of development. These are the 4 criteria that are the most important for us right now.
In 2020, you received nearly $2 million in funding from the nonprofit Global Partnership working to accelerate antibacterial development, CARB-X, to develop therapeutics derived from bacteriophages to kill superbugs and prevent deadly infections in transplant patients. My understanding is you are using phages not to kill bacteria, but as targeting mechanisms to deliver the genetic payloads. Can you explain what you're doing there?
Yeah. So again, we are. It's again we're using the same the exact same approach. We're not phage indeed. We're not using the modalities are not phages, though we are using the phage capsid. So simply the structural component of the phage as a delivery vehicle. In this delivery vehicle, then we have engineered. Now we have methods to package in this delivery vehicle completely synthetic payloads, and indeed, so this vehicle, the particles can be. They have their own targeting properties towards specific bacteria, but because we're really re-engineering them from scratch. We can completely modify their targeting specificities. We can produce them in basically a chassis cell line, which is really cheap to produce. We don't have to produce it in the same type of bacterial species as the bacterial we're targeting, which is still required when people are using phages or engineered phages. And so, yeah, really the capsid for us is the phage capsids is really just a targeting mechanism, but the payload inside this capsid is completely synthetic. It has no element of a phage genome, and therefore the major advantage is that it actually does not replicate, and so we have very beautiful linear dose response to the treatments, and when we administrate the modality either topically or orally.
The company raised $20 million in 2017 from Coastal Ventures and Seventure. How far will existing funding take you, and what's the plan for financing the company from here?
So we're actually just starting the outreach for Series B financing. We're now raising $40 million in order to fund our first human trial with proof of concept in patients, but also the filing of 2 additional INDs and the development of 1 lead candidate in autoimmunity, and other developments, including partnerships, strategic partnership, which are yet undisclosed.
Xavier Duportet, CEO of Eligo Bioscience. Xavier, thanks so much for your time today.
Thank you so much for having me.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send an email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.